Hello, my dear friends. Welcome to Live Prep. Myself, Dr. Gajapati Verma. As I informed you earlier, so under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on our YouTube channel. But the test series can be given only through the app. Clear? And now let us quickly go through the revision of biomolecules in this particular video lecture. Since the chapter biomolecules is little bit bigger, so I am planning to cover this particular chapter in two videos. So here in this first video, so we will go through the basic information that was given under the introduction, and then we will go with amino acids, proteins, lipids, nucleotides, and then nucleic acids. Up to this we will cover in this particular part. So in the remaining part, in the next part, so we will cover carbohydrates, primary and secondary metabolites, and then regarding that chemical reactions also they have given some information. So that part we will cover in the second video. Clear? And before directly starting this particular one, so I wanted to make one more point very clear that in the NCRT under the heading of biomolecules they have mentioned about all these things. But the problem is in the NCRT they have not given information related to one particular thing at one location. Suppose you are talking about the proteins. So regarding the proteins, they have not given entire information at one location. So in different locations, they have given the information. But when we are going for the revision, so if you are studying like that, it will become very difficult. So that's why what I am doing is, so information related to one particular thing. So we are studying at one location at a stretch, so that you can easily understand that particular concept. Clear? And next thing is, so how exactly we are going is, so once we are going with that chemical analysis part, so after that, once we are going with the individual components, first we will study amino acids, then immediately we will go with proteins, so that you can easily understand these particular links and all, because proteins are polymers of amino acids. So these two at a time we will complete, and then we will study about the lipids. Anyhow, we are not observing polymers here, so simply it is an individual component. And then we will study about nucleotides, and then we will study nucleic acids. Nucleic acids are polymers of nucleotides. So these interrelations also you can easily understand. So in this sequence we will try to cover this particular content in this particular lecture. Clear? And now let us quickly go through this. And here, before directly going with the chapter, so here we need to observe one more thing. Under the unit introduction, there are some sentences that are related to biomolecules. Some sentences are related to cell, the unit of life, and remaining are related to biomolecules. But actually they have not given any sentences related to cell cycle and cell division. Already I informed about those things. And now we are going with the sentences that are related to biomolecules. So what is the basic information they have given here? What is the meaning of these particular sentences? All the things we will see here. Clear? So here, so from here onwards, let us see. In studying and understanding the physiological and behavioral process, one can take a physico-chemical approach and use cell-free systems to investigate. So what actually they are saying here? So already regarding these previous sentences, already we covered under cell, the unit of life, right? So those who have not watched that particular one, so you please go through that, and then now you can continue with this biomolecules part. So what we are studying there, so in the case of living organisms in general, if you are talking, different life forms are present, different forms, different appearances are present. Even though organisms are different, cell theory is saying that even though organisms are different, all the living organisms should compulsorily have the basic structural and functional unit called as a cell. So in this way, cell theory has created a mystery, and cell theory is uniting all the living organisms on the basis of the requirement of the living unit that is called as the cell. Yes or no? So this is what the basic information already we have covered. And next here what they are saying, so different organisms are having different physiological reactions that are taking place inside them, and they are showing different behaviors. This also we have studied in the previous, that cell, the unit of life part. And here what they are saying, to understand these physiological processes, to understand behaviors of the organisms, what we need to do? We need to do some investigations. Means we need to do some research. And at what level we need to do? At the micro level we need to do. How exactly an organism is able to show some particular behavior, and what type of reactions are taking place. And to know all these things, we need to know what are the molecules that are present inside a cell. Anyhow, cell is present, but all the cells do not have exactly same molecules. Depending on the location and function, some molecules may be different. And what are those things? So first you need to know about what are the molecules that are present inside the cells. And once you know the molecule, then you need to see what are the functional groups that are present, yes or no, and what are the chemical reactions that are taking place, and what are the products that are formed, and how they are involving in some other process. So all these things we need to understand. Then only we can understand physiological process and behavioral process that are shown by an individual. Yes or no? This is what they are saying. So to investigate all these things, we need to understand all these things. How we will understand these process, and how you will investigate what are the chemicals that are present, what are the molecules that are present. So for that, what they are saying, take a physico-chemical approach. What is this physico-chemical approach? Here they have mentioned, we are using some techniques of physics and some techniques of chemistry. So use the techniques of physics and chemistry so that you can identify what are the molecules that are present inside the cell. This is the physico-chemical approach meaning. And then use cell-free systems. Why we are using cell-free systems? Suppose you are demonstrating a particular process. So what are the components that are required for that particular process? So for that thing, we are using cell-free system. We are not using the complete cell. Only we are using some cellular components, and with the help of that, we are trying to prove that so and so process is happening like this. And in this way, these cell-free systems are useful. So in this way, use physico-chemical approach and also use cell-free systems to investigate all these things. This is what the meaning of this particular sentence. This approach enables us to describe the various processes in the molecular terms. So when you are doing this, you are not just studying as a complete organism. You need to do these particular studies at the molecular level. What are the molecules, what are their structures, how they are interacting, and what are the products that are formed, and how they are responsible for so and so behavior. So these things we need to understand. So at the molecular level, we need to observe this. The approach is established by analysis of living tissues for the elements and compounds. Sorry. So what we need to do? We need to analyze the living tissues. For what? For what type of elements that are present? Nitrogen, carbon, oxygen, like this. What are the individual elements that are present? And not only this, what are the compounds that are present? If you're talking about the compounds, so amino acids are there, lipids are there, nucleotides are there, water molecule is there, likewise many components are present. So in this way, what are the compounds that are present? That also we need to observe. Next one. It will tell us what types of organic compounds that are present in the living organisms. So based on this study, we can understand that what are the various organic compounds that are present in the living organisms. Not only organic, there is a separate analysis, and with that analysis we can do what are the inorganic compounds that are present inside the living organisms also. That also we can understand. Next one. In the next stage, one can ask the question. What is that question? What are these compounds that are doing inside the cell? So first you are saying that, okay, these are the compounds that are present inside the cell. Once you know the list, then what is our next question? What these compounds that they are doing inside the cell? This is what the question. That means how they are interacting. What is the role of those particular molecules? That point we need to understand. And in what way they carry out gross physiological process like digestion, excretion, memory, defense, recognition, etc. So individually, if you are observing, these are the molecules that are present. And ultimately what we need to observe, how these molecules are responsible for various processes like this. This is what we need to understand. This is what they are saying. Next one. In other words, we answer the question. So what is the answer? How we are answering this? What is the molecular basis of all physiological process? The same thing. The same thing in a different way they are saying. So how exactly these molecules are responsible for these physiological processes? This is what we need to answer. Next one. And it can also explain the abnormal process that occur during any diseased condition. If you know about what is a normal condition, in the normal condition, what are the molecules that are present, how they are interacting. If you know that particular information, then you can understand what are the abnormal conditions and how exactly they are happening. in the abnormal conditions, what are the molecules that are formed in abnormal conditions, why they are formed, how they are interacting, how they are responsible for a particular disease condition or some abnormality. So all these things also you can understand. This is what they are saying. Next one, this physicochemical approach to study and understand living organisms is called as reductionist biology. So this type of approach is called as reductionist biology. See, please remember here the word reduction is not with respect to oxidation reduction. It is not that type of reduction. Here the reduction is our view is reduced. Generally we are studying as a complete organism. In that case our view is very broad. Now here we are not observing as a broad view. Our main focus is on a particular molecule or some few molecules. What is the structure, how they are interacting. So in this way our view is reduced. Our view is reduced at a minute micro level we are actually observing. So this type of approach is called as a reductionist biology. This is what they are saying. Very, very important for examination. That's why I am giving the clear-cut explanation. So observe carefully. Next one. The concepts and the techniques of physics and chemistry are applied. That's why it is a physico-chemical approach. Concepts of physics and concepts of chemistry are applied to understand the biology. That's why it is called physico-chemical approach. Next one, in chapter 9, so anyhow, we are talking about some of the biomolecules. Now we are entering into this biomolecules. Clear? I hope now you understood this unit introduction incomplete. Okay? Completely you understood this particular one. Nice. And in the next page of this one, they have given about G.N. Ramachandran, a scientist. Okay, and please remember that scientist is related to biomolecules chapter. Anyhow, now we are studying about biomolecules. So now in this lecture itself we will study about that particular scientist, and that scientist is actually related to protein conformations. In the protein conformations we will study about primary structure, secondary structure, tertiary structure, quaternary structure. Yes sir, in NCERT also they have mentioned. Now out of this, when we are studying about the secondary structure of a protein, and there actually this scientist is related. So that's why what I will do is, now I will not explain about that scientist. So anyhow we are going with the chemical analysis and all, and then we will go with the amino acids, then we will go with the proteins. Yes, in that proteins, when we study about the protein conformations, we will study secondary structure. At that time, I will link up that particular scientist information and what is the relation with this particular chapter. Everything we will study. And please remember, from that scientist information also questions can be asked. I will tell you what type of questions they can ask. Okay, that information also we will cover. And with this introduction is completed. Now we are directly going with the chapter. Anyhow, this is one important one. The physico-chemical approach that is used to study and understand living organisms is simply called as reductionist biology. So in this also they can ask in the examination. So please understand this. Okay. Next one. Now we are directly starting with the chapter, that is the biomolecules. So information is very clear and simple, and please remember this chapter has huge information. If you want to discuss, you can discuss so much of information, but please remember, since it is a revision lecture, I am simply sticking myself to the NCERT content. That's it. Okay, just for some of the important things, some 1 or 2 important points I will add it, and some interlinked points are also there. Those interlinked points also we will discuss. Clear? So now mainly we are focusing on NCERT content only, and as I informed you earlier, information related to one particular content we are studying at one location. Even though in NCERT they gave in different locations, at one location we are covering that complete information. Clear? Now let us quickly start here. So biomolecules, here first initially what they are saying, whatever the elements that are present within the earth crust, the same elements are even present in the living tissue also. That means the elements in non-living thing like earth crust and even in the living cells, all these elements are exactly same. Even though elements are exactly same, if you are calculating the proportions may be different. Proportions may be different. If you are talking about the earth crust means what? It is about the soil. Soil is silicon dioxide, SiO2, yes or no? Soil is silicon dioxide. It is SiO2. So obviously you can expect that silicon as an element, if you see, silicon is present and oxygen is more present. So in the earth crust, if you see, silicon and oxygen are more present. But in the case of living organisms, if you are observing, so here generally when compared with the earth crust, if you are seeing, so carbon and hydrogen are more in proportion in comparison with non-living. Yes or no? So they are more in living organisms in comparison with non-living thing like earth crust. This is what another sentence that they have given in NCERT. And then we are going with how to analyze this chemical composition. What is the exact procedure? So even though I have given like this, now I will try to write or explain with the help of a diagram. Okay. Now let us quickly go through this. So initially they are taking a tissue. Tissue means what? It is a group of cells. Suppose it is a tissue. We are taking a tissue. Any tissue we can take, plant tissue, animal tissue, or even a microbial paste also we can take. Anything you can take. So some group of cells we are taking. Now what we need to do? We need to break the cells. We need to break the cells so that we can extract the components. So here what we are doing, we are taking this particular tissue in a beaker, and to this we are adding an acid. What is that acid? It is trichloroacetic acid. What is normal acetic acid? CH3, CH3, COOH is called as acetic acid. But here what we are using? We are not using just acetic acid. It is trichloro, means 3 chlorines. Where these 3 chlorines are present? They are replacing these 3 hydrogens. So it is a trichloroacetic acid. This is what they are talking. And now you can understand this chlorine more electronegative, yes or no? That means, please understand here, there is one logical point is there. This logical point is linked with the lipids. Why lipids are coming in macromolecular fraction? There is a logic here. So please observe here. This is electronegative. So that means here there is some polarity that is present here. So that's why it is a polar solvent. It is a polar solvent. I am giving the direct conclusions. I don't want to go into individual informations. So the solvent that we are using is trichloroacetic acid, and this trichloroacetic acid is a polar solvent. Please remember this, polar solvent. Now we are adding this trichloroacetic acid, and then we are breaking these cells. Once you are breaking these cells, now what happens? These components are released into this acid. These components or the individual molecules are released into that trichloroacetic acid. So in this way we are making one particular semi-solid paste like thing we are preparing. Now this is ready. And then what we need to do? After this, now we are taking a separate beaker. We are taking a separate beaker, and on this beaker we are keeping one cheesecloth. Cheesecloth you can keep it, or cotton also you can keep it. Suppose it is a cheesecloth. For understanding purpose, we are taking like this. It is a cheesecloth. Now you just imagine a cloth. I am going with internal points also, so please observe carefully so that your concept will become very easy. This is a cloth. Cloth means what? It is actually made up of some fibers. Yes or no? And between the fibers there are some spaces. There are some small pores. P-O-R-E, pore. There are some small pores that are present in this cloth. Even in the cotton also, if you are taking, what happens? There will be some small pores that are present between that. So now the thing is, these pores are very, very important. Now what you are doing? You are taking this mixture and you are placing on it, and then you are pressing it. Now what happens? Now what happens? There are so many molecules that are present in this. Some are small molecules, some are large molecules. Yes or no? And that too, the solvent acid is present. Now in this way, now this entire content we are keeping on it. Now what happens? Acid is able to pass through this. Acid is able to pass through this, so that now acid is collected inside the beaker. Acid is collected inside the beaker. This is the acid. This is the acid. Now, along with the acid, some small molecules are coming. Why small molecules are coming? Because this cloth is having a particular pore size. Small pore size is there. The molecules that are smaller than this pore size, they can easily pass through this. So they are coming through this particular pore and coming into the acid. So in this way, acid plus some small molecules are present. Some small molecules are present. Yes or no? This is what. And these small molecules are called as micromolecules. These small molecules are called as micromolecules, and they are present in the living system. So that's why they are called as biomicromolecules are present. So they are the small molecules present in living systems. That's why they are called as biomicromolecules. So in this way, what is the overall conclusion? So in this way, when we are filtering this particular paste through this particular cheesecloth, then what we are observing? Acid along with some small molecules is collected inside the beaker. Understood or not? So upon filtration, we are getting this. That's why this particular fraction is called as filtrate. This particular fraction is called as filtrate. So what are actually present in the filtrate? Filtrate is actually having the acid, trichloroacetic acid, along with some small molecules. This is what having. And now what is the other point? Now here some molecules that are smaller, they are coming into this. But what about the molecules that are larger in size? Suppose there are some molecules that are larger in size. Can they pass through this particular pore? No, they cannot pass through. Then what happens? Now all these molecules, they are collected at the top of this particular cheesecloth. Since these are large molecules, they cannot pass through the small pores. That's why all these are stopped on the top of this particular cloth. And this particular fraction is called as retentate. This particular fraction is called as retentate. Please understand these names also. Filtrate and then retentate. And what actually the filtrate is having? Acid along with small molecules called biomicromolecules. But what about the retentate? Retentate is having large molecules. So that's why these are called as biomacromolecules. 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 So please observe all these points. So this is what it is actually having. Next important point that we need to know is, so since these are small molecules, so what is the average size of these particular molecules? This is also important. So generally they are having the size range of 18 to 800 daltons. 18 to 800 daltons. Or else you can also say this as less than 1000 daltons. In NCRT, in one sentence they used this, in another sentence they used this particular one. Both of them are correct, so no problem. So those molecules that are having the size less than 1000 daltons, they are coming along with the acid, and these are called as biomicromolecules or simply micromolecules. That's it. But what about these macromolecules? Generally here the size of these macromolecules is more than 10,000 daltons. More than 10,000 daltons. So instead of writing 10,000, you can also write this as more than 10 kilodaltons, small k. That kilo, whenever you are writing kilo, it should be small k, kilodaltons. So this is what generally they are saying. So this is what some general information that we need to understand with respect to this filtrate fraction and retentate. Clear? Next important point. What is the next important point? Next important point is what type of molecules are present. Are they organic only, inorganic only, or both? This is also very important point. Many students are not focusing on this particular point, so observe carefully. Filtrate fraction. Filtrate, this is having what type of molecules? I am giving you options. Only organic, only inorganic, both. Very important question. Filtrate. Filtrate is also called as acid soluble pool. It is also called as acid soluble pool. Acid soluble pool. And the retentate is also called as acid insoluble pool. Acid insoluble pool. You need to know all these names. Names are very important. Filtrate is also called acid soluble pool. What are the components that are present in this? It has both organic. It has both organic molecules and inorganic molecules. Both organic molecules and inorganic molecules. Very important. Observe carefully. Then what about the retentate or acid insoluble pool? It has only organic molecules. It has only organic molecules. It does not contain inorganic molecules. Please remember, in the living organisms, inorganic polymers are not present. Only organic biopolymers are present. Clear? So please understand this. So filtrate is having both organic and inorganic components, but retentate or acid insoluble fraction is having only organic components. Understood or not? That means the same information can be said in a different way. What is that? Some organic compounds are present in the filtrate and some organic compounds are present in the retentate. Can you say like this or not? Yes, 100% you can say like this. What is the other sentence for inorganic? All the inorganic components, please understand, in the living systems, all the inorganic components will come under acid soluble pool, that is a filtrate only, and inorganic components are not present in retentate. And this is the information, very important for examination. So that point you need to observe very carefully. Clear? So in this way we are talking about this general information. Yes. And with this general information is completed. And then we are going with what are these components now. Okay, you are saying organic here, and inorganic here only. Nice. But what are these components? So now we will write some of the important examples. What are these important examples? So what are the organic compounds that are present in the filtrate? These are all small organic molecules. These are all small organic molecules. And what are these? Like some simple sugars like glucose, sucrose, all these things, small molecules. And not only this, amino acids, individual amino acids, they are present in the acid soluble pool. Not only this, individual nucleosides, nucleotides, nitrogenous bases, they are also present within this acid soluble pool. So in this way, some small organic molecules are present in this acid soluble pool. And regarding the inorganic molecules, some calcium carbonate, some phosphates, likewise some inorganic components are also present within this acid soluble fraction. Clear? And now our major focus is acid insoluble pool or retentate. What are the organic molecules present in this? And what are the organic molecules present in this? There are 4 types of organic molecules present in this. 4 types of organic molecules present in this. What are those? First one is proteins. First one is proteins. Proteins. Next one, nucleic acids. Nucleic acids. Nucleic acids. And the third one is polysaccharides. 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 And the last one is lipids. And the last one is lipids. Last one is lipids. So please remember, in this retentate or acid insoluble fraction, that means here what are present? 4 types of organic. All the 4 are organic only. 4 types of organic components. What are these? If you are classifying them, you will find proteins, nucleic acids, polysaccharides and lipids. Only these 4 types of organic components are present in acid insoluble pool. This is the overall information regarding this particular process. Clear? And now for the examination point of view, so now I will tell some basic information. Try to understand carefully. Proteins are present in retentate. Proteins means what? They are polymers, right? They are the long molecules, polymers. So they are present within this retentate. But what is the individual monomer is called as? What is the monomer for the proteins? Mono means single, mer means unit, single unit. So proteins are polymers of amino acids. But where you will find individual amino acids? Individual amino acids are present in the filtrate, but proteins are present within the retentate. Observe this carefully. Next one, nucleic acids. Nucleic acids are also polymers, so that's why they are coming under retentate. But what is the individual unit is called as? One single unit is called as nucleotide. And where you will find the individual nucleotides? Individual nucleotides are seen in the filtrate. Please observe carefully. Next one, polysaccharides. Poly means many sugars. Many sugars are attached to each other. This is also a polymer. This is also a polymer, and this polymer is observed in the retentate. But what about individual unit? Individual unit is called as monosaccharide. And where you will find monosaccharides? In the filtrate. Please observe carefully. So in this way, for these 3 first we will see. For these 3, if you are observing, these 3 are actually true polymers. These 3 are true polymers. True polymers. Why they are true polymers? Because many units are attached to each other. Many units are attached to each other. In this way, these 3 are true polymers, and these 3 polymers are present in retentate. But their individual units, single units, individual units are called as monomers, and they are seen in the filtrate fraction. Clear? Now this last one is very important. Lipids. Are they polymers? They are not polymers. Lipids are not polymers. These are not polymers. These are not polymers. If they are not polymers, we will not talk about monomer. So that's why there is no monomer concept here. So they are not polymers. And that too, what is the size of these lipids? See, in the general size, what we are saying, more than 10,000 kilodaltons. So more than 10,000 kilodaltons is applicable to these 3. But for the lipids, what happens is, if you see individual lipid molecule, its size is less than 1000 dalton. Less than 1000 dalton. More specifically, it is less than 800 daltons. It is less than 800 daltons. Understood or not? That means, technically, if you see, what should be the position of lipid? Since it is a small molecule, lipids are less than 800 means what? They are small molecules. If they are small molecules, where actually we are expecting their location. So they should come under the filtrate or acid soluble fraction, they should come. But actually where we are observing? We are observing under acid insoluble pool, that is retentate. What is the logic behind this particular one? Very clearly they mentioned in NCERT. What is the reason here? I am giving the explanation first, then we will read the NCERT sentences. Don't worry. What is the reason for this? The reason is the acid. What type of acid we are using? Polar solvent. The acid trichloroacetic acid is a polar. Lipids are non-polar. Lipids are non-polar. Please observe carefully. The acid that we used in this particular process is a polar solvent, but the lipids are non-polar. Now you just observe carefully. Can these non-polar lipids solubilize within this acid, polar acid? No, they are not solubilized. This is the reason. So since these are non-polar and the solvent used is a polar solvent, now these are not miscible. They are not miscible in this particular acid. If they are not miscible, what happens? Now all these non-polar lipids, now they are coming together. Once they are coming together, now what they are doing? They are making some large vesicles. They are making large vesicles like micelles they are forming. And since, please remember, individual molecule is small molecule, but once they are grouped together, they are forming large vesicle. And this large vesicle cannot pass through this small pore. That is the reason why they are present along with this macromolecules. But originally they are not macromolecules, they are not polymers, they are not macromolecules, they are not true macromolecules, they are not true polymers, but still they are present along with macromolecular fraction because they are non-polar and the solvent used is a polar solvent. That's why all the lipids will gather together, come together, and they are forming vesicles or the micelles so that their size is increased now. And now these vesicles cannot pass through this particular cheesecloth. That's why they are present along with this. But originally they are not polymers and their individual lipid size is also lesser than 800 Daltons. Very clearly they gave all this information. Okay. But where these lipids are present? Lipids are present in the membranes. Yes or no? They are part of the membranes. So various membranes are present. Already we studied in the cell chapter. So that's why all these lipids are coming out and they are gathering together and they are forming the vesicles. And this is what some basic information that they have mentioned in the NCERT. Clear? I hope now you understood this. You need to remember these names and everything, where they are present, which fraction, whether organic or organic plus inorganic, all the things you need to observe carefully. Some very small basic information. Clear? Now come to this. This is what the process. I have explained this very clearly. Next one. So then what we need to do? So once in this way you are getting some compounds in these particular fractions. Then what we need to do? Now separate all these components from each other and then go with some chemical and physical techniques, and finally you can get the structure of these particular components. Clear? This is what they are saying. So in this way, once you get the structure, you can make the formula and all the things so that we can use that particular information for understanding what are these molecules that are present, what is the nature, what are the chemical groups that are present, and how they can interact with each other, what are the different reactions that they are doing, and what type of products can be formed. All the things we can understand from this particular one. So that's why before understanding all these things, first thing what we need to do, we need to isolate these particular molecules and get the structure. That's what they are saying here. Clear? In this way, so all the carbon compounds that are present within these living organisms or living tissues or living cells are simply called as biomolecules. These are the molecules that are present within living systems in simple. Clear? These are called biomolecules. That is for in general. But here in NCERT, separately they are mentioning about how you will analyze inorganic elements specifically. How you will analyze the presence of inorganic elements and what type of inorganic elements are present, what type of inorganic components are present, and how you are doing this particular analysis. The process is very simple. Initially you need to take some living tissue. Living tissue means generally it is having more water in it. If water is present in it, then it is called as a wet weight of a tissue. So that's why first you need to dry it so that water will evaporate. So once water is evaporated, then once again we will measure the weight, and then it is called as a dry weight. And now this particular dried tissue we need to take it and then you just burn it. Once you are burning, what happens? All the carbon compounds will be evaporated in the form of water and CO2. Please remember, burning of living tissue will result in removal of carbon compounds, organic compounds. All the organic compounds will be removed. And how they are removed? Generally in the form of water and then CO2. So in this way, from the tissue, you have removed organic compounds. Once you are removing organic compounds, then you will get some powder. That powder is called as ash. That powder is called as ash. And what this ash actually consists of? This ash actually consists of inorganic components. So in this way, that ash you need to collect and then go for the analysis of these inorganic elements. And you will get the inorganic elements, compounds and everything, entire list you will get here. So in this way, from a tissue, you can also analyze about what are the various types of inorganic compounds that are present. And then you can make a big list of this. So here in the NCERT, they have given one table. In this table, they are comparing between earth crust and the human body. And here they are talking about what are the various elements that are present and the percentage weight with respect to these particular components also they have given. So observe these percentages. So in the earth crust, what are the elements that are more present by percentage? In the living organisms, what are the components that are more present? And not only this, and here how many times it is more present in human body? Or reverse, if you see how many times it is more present in earth crust, like that also questions can be asked. So please analyze this particular table. Proper analysis we made in the regular lectures. So if you are more interested, you can go through those regular lectures also. And then here there is a list of these components with respect to this inorganic components. So what are the various types of ions that are present? What are the various compounds that are present? Some big list also they have given. So please go through this particular list also. Next one. So once we are going we are getting these individual components. So we are getting finally the structures. Mainly chemistry point of view, if you see, simply we are observing with respect to what is the molecular formula and what is the functional groups. But in the case of biological point of view, instead of talking with respect to functional groups, and here we are segregating or classifying them into various names like amino acids, lipids, nucleotides, proteins. Likewise, we are using different names. Okay, so this is what they are saying here. Next one. So this is another comparison that they have given in a different location, but we are writing here itself. Acid soluble pool, it represents roughly the cytoplasmic composition. Majority of the cytoplasmic components are present within the acid soluble pool, but please remember some proteins, carbohydrates like this, they will come under this acid insoluble pool. That's why they are saying that it is roughly represent cytoplasm. They are not saying that entire cytoplasmic components are coming under acid soluble. Majority of the cytoplasmic components are coming under acid soluble pool. And here the size is approximately between 18 to 800 Daltons. That means less than 1000 Daltons is the size of these molecules. And these are called as biomicromolecules, or simply they are called as biomolecules. Size is less than 1000 Daltons. And different components like individual small sugars, amino acids, nucleotides, inorganic compounds, all these things will come under acid soluble pool. Next one is regarding acid insoluble pool. It is also called as retentate. Acid soluble is called as filtrate. Please remember the names also. Acid insoluble pool or retentate, and here the molecules are generally they are having the size greater than 10,000 Daltons, 10 kilodaltons. Next one, but the exception is lipids. Okay, next one. So these are simply called as biomacromolecules, large molecules, and the proteins, nucleic acids, polysaccharides, and lipids are present. Okay, all these are polymers except the lipids. So lipids are not the polymers, but proteins, nucleic acids, and polysaccharides are polymers. Clear? Next one. Next one, they are talking that, so why these lipids are coming under the macromolecular fraction even though their size is less than 800. Already we studied this. Solvent used is a polar solvent. Lipids are non-polar. So that's why they are not miscible in acid. So that's why they are grouping together, forming the vesicles, so that the vesicle size is larger than pore size of that cheesecloth. So that's why it is coming along with the macromolecular fraction. Very clearly they have given this particular information in the NCERT. So that's why lipids are not strictly macromolecules. Next one, water is the, if you are observing the chemical analysis and all, what they are saying here, water is the most abundant chemical in the living organism. So overall all the chemicals, if you see, water is the most abundant chemical, but water is organic or inorganic? If you are talking like this, water will be inorganic. So, and remaining components, protein, carbohydrate, lipids, nucleic acid, ions they have given. So forget about the ions. Now these four, let us see. So proteins, carbohydrates, lipids, nucleic acid, these four will come under organic components. So what is the most abundant organic compound if they ask? Then you should not write water. Organic compound directly if they are asking, then you should not write the water. Then what you need to write? Out of these four, what are the most abundant? Proteins are the most abundant. 10 to 15% are present. Yes or no? So in this way, in general, if they ask, water is the most abundant. Inorganic, if they are asking, water is the most abundant. And organic, if they are asking, proteins are the most abundant. So read the question very carefully. Clear? Now, out of these four also, they may ask you to arrange based on the percentage. Now, proteins 10 to 15%, nucleic acids 5 to 7%, carbohydrates 3%, lipids 2%. So, they may ask in ascending order or they may ask in descending order. So, you need to remember them in a one particular order. And these values also you need to remember because they have given in NCERT directly. These values also they may ask in examination. So, please be careful. Please be careful. Don't ignore anything. Any information given in NCERT is important for examination. Now let us go with the amino acids. Now till now that chemical analysis part is completed. Now we are going with individual components. Already I told you. First we will study about amino acids. After completion of amino acids we will go with proteins because proteins are polymers of amino acids. So these two informations will be linked with each other. So it is easy for you. So that's why now we will go with this. Here also we are strictly going with NCERT information only. So please observe carefully. Amino acids. These are the molecules that are having at least one amino group and one acid group, COOH, carboxylic acid group. So that type of molecules are called as amino acids. So any molecule having at least one amino group and one carboxylic acid group, that is called as amino acid. Please remember, at your level you are studying about 20 standard amino acids. What are standard amino acids? Standard amino acids are the amino acids that are introduced into the protein through the process of translation, and only these are called as standard amino acids. At this level of class 11th and 12th, for the NEET UG examination, you are studying that standard amino acids are 20 in number, 20 in number. Why I am saying like this? Because in higher classes you will study about some extra standard amino acids. So there are some amino acids like selenosystine, pyrrolysine. Likewise, they are also standard amino acids but not mentioned in NCERT. So for our level it is 20 in number. So only those 20 only you need to observe carefully. And what are these? Out of the 20 in the NCERT they gave total 3 amino acids names only. In general, if you are observing in these amino acids, all these 20 standard amino acids are alpha amino acids. That means alpha carbon is having amino group. Alpha carbon is having amino group. If you are observing this, COOH is the first one. The carbon attached to COOH is the alpha carbon. On the alpha carbon, amino is there. Alpha carbon, amino group. Alpha carbon has amino group. That's why they are called as alpha amino acids. Clear? And you can also write these amino acids as substituted methanes. What is methane? CH4. CH4 is the methane. So carbon having 4 hydrogens here. That is methane. If these hydrogens are substituted with some other groups like this, now you can call these as amino acids. So that's why amino acids are generally considered as substituted methanes. Now here, one is COOH group, that is compulsory. One should be NH2 group, and other third one is hydrogen. Up to these three are fixed, and the fourth one is variable. So that's why generally we write it as R group. Variable side chain we will write it. So based on this, if that R group is hydrogen, if that R side chain is hydrogen, now this amino acid is called as glycine. If that R group is CH3 methyl group, it is called as alanine. If that R group is CH2OH, means hydroxy methyl group, then it is called as the serine. At least just these three structures they have given in NCERT. So please remember this. So in this way, these amino acids are the organic compounds and all these are alpha amino acids, and these are considered as substituted methanes. Clear? Next one. So the same information I have written here, but actually you need to talk about total 20 standard amino acids. In the regular lecture we have studied what are all these names and everything. So if you are interested, so please go through that. Next one. So what are the chemical and physical properties? So these amino acids, generally in the biology, under the biomolecules chapter we are not studying this, but actually in the chemistry also there is a biomolecule chapter. There you are studying about different types of reactions and all, right? So this sentence is actually related to that particular chapter. So chemical and physical properties of amino acids, they are mainly due to, I am not saying that only, mainly due to what? This R functional group. You are talking with respect to R functional group. But not only this, Even amino and carboxyl groups are also involving in the reactions. So that's why the chemical and physical properties of amino acids are due to amino group, carboxylic acid group, and even the R functional group, side chain, R chain, that is also involving in the reactions. This is what they are saying. And next one, based on the type of this R group, based on the nature of this R group, we are classifying amino acids into different types. So some amino acids are called as a basic amino acids, and what type of charge they are having? They will have the net positive charge. And here, what are the standard examples here? Lysine and arginine. These are the two examples that you are studying, but along with this, histidine is also there, that is also basic amino acids. So please remember this. Next one, acidic amino acids. So they are having acid group extra in the side chain. So side chain is also having one extra COOH group. That's why these are negatively charged. These are called acidic amino acids. What are the examples? Aspartate, also called as aspartic acid. Both of them are same. Next one is glutamate, also called as glutamic acid. Both of them are same. So in this way, aspartate or aspartic acid, glutamate or glutamic acid. So these are the two examples for acidic amino acids. Next one, aromatic amino acids. In the side chain, aromatic group is present. So what are the examples here? Tyrosine, tryptophan, phenylalanine, and even histidine is also aromatic. So these are the four examples from the NCERT that you need to remember. Clear? Next one, hydroxyl group. Hydroxyl group in the side chain, if you are talking separately in the side chain also, if OH group is present. So what are that type of amino acids? They are serine and threonine. Please remember this. Next one is regarding sulfur containing. In the side chain, some amino acids are having the sulfur. They are cysteine and methionine. Cysteine and methionine. Please remember these two names. In the molecular basis of inheritance chapter, we are studying about Hershey and Chase experiment. In the Hershey and Chase experiment, so there we are using this radioactive sulfur. Radioactive sulfur. So this radioactive sulfur used in that experiment will go to what type of amino acids? Sulfur containing amino acids like cysteine and methionine. This is one interlinked information. So you need to compulsorily remember this. Clear? Next one. Next one is regarding secondary amine containing. What is secondary amine is called as imino group. Please remember, generally we are saying about amino acids. Amino group, NH2 group is present. NH2 group is present generally. But please remember, out of 20 amino acids, 19 amino acids are having primary amine. What is primary amine? NH2 group. This is amino group. Amino group containing amino acids are 19 amino acids. 19 amino acids. But one amino acid is having imino group. Imino group. This is called as a primary amine. This is called as primary amine, but imino group is secondary amine. And this is having NH like this. It is having like this is called a imino group, but this is also a standard amino acid. That's why you are studying this under amino acids list. So in this way, 19 amino acids are having amino group, whereas one amino acid is having imino group. And what is that secondary amine or imino group containing one? This is the proline. Proline is the standard amino acid that is having secondary amine, also called as imino group. Please understand this. Clear? This is also another important point, but not mentioned in NCERT, but better remember this. This question can be asked in chemistry also. So please observe this. Next one is regarding some hormones information. So there is a separate chapter called as chemical coordination and integration chapter. So in that chapter you studied about some hormones. But please remember, some hormones are derived from amino acids. What is this list? This is also very important. So you need to compulsorily remember this. Epinephrine and norepinephrine, they are also called as adrenaline and noradrenaline. They are produced from adrenal glands. Yes or no? Adrenal medulla is producing this. And now these hormones are derived from tyrosine amino acid. So from the tyrosine amino acid, these hormones are synthesized. Next one, thyroxine. Thyroxine is produced from thyroid gland, and it is also produced from tyrosine. From the tyrosine amino acid only it is produced. And here you are studying about, from the thyroid gland you are studying about T3 and T4, right? So this one only, this is the T4. So here T3, triiodothyronine, T4 is tetraiodothyronine. So both of them are derived from tyrosine amino acid only. So please remember this one. Next one is regarding melatonin. Melatonin is produced from the pineal gland, and this is produced from tryptophan amino acid. This table is very important for examination, so please remember this. This is interlinked information. Clear? Next one, they are saying that in solutions of different pH, the structure of amino acid changes. What is the meaning of this? If the pH of solution changes, structure of amino acid changes. How? I will explain you. In the NCERT, they have given this. Quickly, I will try to explain you the same diagram so that you should never make any mistake. And please remember, this concept is important even in higher classes also. So I am explaining very clearly. So please try to understand this. Here we are talking about total 3 conditions. What are these 3 conditions? The condition at which pH is very less, and here the condition where pH is very high. Understood or not? pH is very less and here pH is very high. Very high. And now observe carefully. pH value is high means H plus, sorry, here pH value is low means H plus ion concentration is more. And here pH value is high means H plus ion concentration is less. Yes or no? This is what the logic is, and this is what. And here we are talking about intermediate, another one. So slowly I will explain. First we will talk about a condition where pH is less. pH is less means H plus ion concentration is more. That means in this solution H plus ion concentrations are more present. If H plus ions are more present and in that solution if amino acid is present, now what happens? This H plus will go and attach to amino acid. Clear? First I will write the normal structure of amino acid. This is carbon, these are the 4 groups. Suppose this is COOH, suppose this is NH2, and suppose this is H, and this is R side chain. And please remember, actually R side chain is also important, but at this level of class 11th and 12th, you are not focusing on R side chain. In higher classes you will study about that. And here our focus is only COOH group and NH2 group, that's it. Only these 2 things we are observing. Now let us see. Suppose, for understanding purpose, you are taking the structure like this. If this is the basic structure of amino acid, then at very low pH, since H plus ion concentration is more, that means inside the solution more H plus is there. Now what happens? That H plus will go and attach to this NH2 group. H plus will go and attach to NH2 group. So now it will become NH3 plus. If H plus is attached, now what happens? It will become NH3 plus. That means in the solutions where pH value is very less, what is the structure of amino acid? This is the structure of amino acid. And in this structure, what is the net charge? What is the net charge of amino acid? What is the net charge of amino acid? Here it is plus. It is the plus 1. How many plus are there? 1 plus is there. Only 1 is plus. So I am not talking about R group. So based on this, how many plus groups are there? Only 1 plus is there. So it is plus 1. Right, like this. So net charge of this particular amino acid is positive. Net charge is positive and the value is plus 1. Nice. And when you are slowly increasing the pH, here pH is very less. Now suppose here pH is 2 and now you are slowly coming to 3, 4 like this. So when you are slowly increasing the pH, now what happens? pH is slowly increasing means H plus ion concentration is slowly decreasing. When H plus ion concentration in the solution is decreasing, now what happens? Amino acid will try to give its H plus. That means amino acid will lose its H plus. Now observe carefully. Who can donate H plus? COOH can donate H plus and even NH3 plus also can donate H plus. But between these two, who is more acidic? Who is ready to give H plus first? COOH group is more ready to give H plus. So when your pH is slightly increased from this, then what you are observing? A COOH is donating it H plus. So now what happens? COO minus it will become, but the remaining structure will remain same, and this will be like NH3 plus and this is R group. Understood or not? Now what is the net charge? Here it is plus 1, this is minus 1. Plus 1, minus 1. That means what is the charge here? Net charge is 0 here. Net charge is 0 here. So the structure of amino acid where number of positive charges is equal to number of negative charges, or the structure of amino acid where net charge of amino acid is 0 is called as Pi. What is this? Pi means? I means isoelectric. P means point. It is called as a Pi. It is the, what is pI? pI is a particular pH value. The pH value at which the isoelectric point means what? So the number of positive charges is equal to number of negative charges, or net charge of amino acid is 0. That particular pH value is called as a pI. So please remember, different amino acids will have different R groups. Different amino acids will have different R groups. So R group is also playing some role. That's why different amino acids will have different pI value. Different amino acids will have different pI value. That means at that particular pH value, for that particular amino acid, the net charge will be 0. Number of positive charge will be equal to number of negative charge. So in this way, every amino acid will have its own specific pI value. Clear? And this particular structure is called as a Zwitter ionic form. It is called as Zwitter ionic form. So this is what. So the structure of amino acid where number of positive charges is equal to number of negative charges is called as Zwitter ionic form. Very, very important one. So please observe carefully. Suppose the pH value is further increased, then what happens? If it is further increased, now what happens? In the solution, pH is increased, means H plus ion concentration is further decreased. In the solution, H plus ion concentration is decreased, means now what happens? Already COOH donated H plus. Now who will donate? NH3 plus will donate. So now what happens to the structure here? It will be like COO minus, and this will be H, this will be R group. But here what happens? Instead of NH3 plus, from the NH3 plus, H plus is donated into the solution. That means it is lost. Now what happens? It will be existing like NH2. It will be existing like NH2. Now what is the charge on this amino acid? It is negative charge. How many negative? In this structure it is -1. It is -1. Understood or not? So this is how, this is what they are saying. In solutions of different pH, try to understand NCERT sentence first. What they are saying here, in solutions of different pH, the structure of amino acid changes. How it is changing? Now observe carefully here. So this is the solution of different pH. In solutions of different pH, different structures are present for amino acid. This is what they are saying. So this concept is very, very important. Even in higher classes also, many students are making mistakes. So please observe carefully. So what are the overall conclusion? At very low pH, what is the charge on amino acid? It is a positive charge. At very high pH, what is the charge on amino acid? It is negative charge. The pH at which net charge of amino acid is 0 is called as isoelectric point, indicated with Pi. Please observe this. This concept is very, 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 very important. Very, very important. So please observe it carefully. Don't try to by heart this. Try to understand the logic. How exactly it is getting changed. So in here, what is this A condition? This is very low pH. What is this C condition? Very high pH. What is this B? This is the Pi, isoelectric point condition. So that's why at this condition, whatever the structure that you are observing, it is called as Zwitter ionic form. Understood or not? This is some basic, very important information. With this NCERT information regarding the amino acids at this location is completed. Now at different location. At different location, they have mentioned about essential and non-essential amino acids concept. What is this essential and non-essential amino acids? Please remember, our body can synthesize some amino acids. Our body can synthesize some amino acids, but other amino acids are not synthesized. So in this way, what happens? Those amino acids that are produced by our body, anyhow our body is producing. So you don't need to take them externally. Externally through the diet, you don't need to take them. So that's why they are called as non-essential amino acids. So the amino acids that are produced by our body are not essential amino acids, but those amino acids that are not produced by our body is not producing, but our body requires them. Then how you will get them? Through the diet, through the food. So that's why those amino acids are called as essential amino acids. So let us go through once again. The amino acids that are not produced by our body are essential amino acids, whereas those that are produced by our body are non-essential amino acids. This is what the list is. Clear? So what are these? Essential amino acids. So MATTVILPHLY. This is how we have written. Mat will fly. This is some short thing that you can understand. What are these? Methionine, arginine, tryptophan, threonine, valine, isoleucine, leucine, phenylalanine, histidine, and lysine. Generally, if you are considering this, these are the 10 amino acids that will come under essential amino acids list. In this also, if you observe, arginine, that means from this side second one, from this side second one. So arginine and histidine, generally these are considered as semi-essential. Why you are talking about semi-essential again here? So why they are semi-essential? Because in the adult individual they are not essential. In the adult they are not essential, but in the children they are essential. So that's why these are considered as semi-essential amino acids. But this list of this particular thing was not mentioned in NCERT. Clear? But simply the names they have mentioned, at least that information you please remember. Clear? Now with this, information regarding amino acids is completed. Now we are directly going with the proteins. What are these proteins? Proteins are polymers of amino acids. Amino acids will attach to each other so that they are making a long polymer called as a protein. This is what the important point is. So proteins are polypeptides. Many amino acids will attach to each other. That's why they are called as polypeptides. Next one. So they are the linear chains of amino acids. Amino acids will attach to each other. So that's why they are called as linear chains of amino acids. Next one, they are linked by peptide bonds. What is the bond that is connecting two adjacent amino acids? It is called as a peptide bond. Biologically, it is called a peptide bond, but chemically it is called as amide bond. So please understand this. Peptide bond is also called as amide bond. So please remember this. Next one, these are heteropolymers. They are not homopolymers. What is the meaning of heteropolymer? Different. Hetero means different. So different amino acids are present. It is made up of different units. Different unit means what? So different amino acids are present. So that's why proteins are heteropolymers. And here in the NCERT, they have given one particular table. So here they have given some name of the protein and its function. Collagen protein is acting as intercellular substance, intercellular ground substance. Between the cells, it is present in the tissues. In the case of animal tissues, you are observing this. Next one is a trypsin. Trypsin is the enzyme that you are studying under the digestion and absorption chapter. So though it is deleted, so since this name was mentioned, at least remember this. So this trypsin is a protein digesting enzyme. Okay, next one, insulin. Insulin is a protein that is acting as a hormone. In the chemical coordination and integration chapter, you are studying about this. Next one is regarding antibodies. Antibodies are also proteins. They are also proteins. In human health and diseases, we studied about the structure of this antibody, right? Two heavy chains, two light chains, and they are interlinked with some disulfide bonds. All the things we study now. What are those chains? At one side N-terminus, other side C-terminus. What are these? Again, they are proteins only. Next one, receptors. Not all receptors, but majority of the receptors are made up of the protein. And these receptors are present on the surface of the cells, and they are having different roles. Some are having sensory reception like smell, taste, hormone. Not only this, some protein receptors are involving in cell-to-cell interaction, recognition. Many things are there. Many things are there. They are just some small examples. Next one, GLUT4 is a protein. Directly, it is an example they have given. So GLUT4 is a protein, and what it is doing? GLU, glue means glucose. T means transport. So it is helping in glucose transport. So that's why enables glucose transport into the cells. From the outside, the glucose is entering into the cells. For this particular transport, this particular protein is important. So this entire table also you need to remember. Next one. Next one, they are saying that collagen is a protein that is most abundant in the animal world. Only in the animal kingdom, if you are observing, within the animal kingdom, if you see, the most abundant protein is collagen. But if you are talking about entire biosphere, then the answer will be rubisco, ribulose, 1,5-bisphosphate carboxylase oxygenase. And where we are observing this? In the plants. So if they are talking about plant kingdom, then the answer will be rubisco. If they are talking about entire world as a earth as a biosphere, if you are observing, then also it is rubisco. But if they are asking only for the animal world, then only the collagen will be the answer. Most abundant protein will be the collagen. Clear? Next one is regarding structure of the protein. This was given at a different location. We are combining all this information at one location. Structure of the protein. So proteins are heteropolymers and they are made up of the amino acids. And here we are studying about the different conformations. In the NCERT, they said that in the case of chemistry, generally they are focusing on in the case of inorganic chemistry, it is just the molecular formula that will give the structure. But in the case of organic chemistry, we are drawing about the three-dimensional, yes or no, some structures in the two-dimensional way we are representing. Generally in the physics, we are using the three-dimensional representation. But in the case of biology, we are using four levels of organization for this particular protein structure we are studying. Don't say four dimensional, four levels we are saying. What are these levels? It is the primary structure, secondary structure, tertiary structure and quaternary structure. So primary structure, what actually the primary structure? It is simply the sequence of amino acids from N terminus to C terminus. What is N terminus? Amino terminus. To C terminus means carboxy terminus. Please remember, for the first amino acid, amino group is free. For the last amino acid, carboxylic acid group is free. And please remember, during translation, what is translation? Protein synthesis from mRNA, right? So during protein synthesis, the amino acids are attached in this sequence from N terminus to C terminus. That's why always the reading of protein is from N terminus to C terminus. So that's why simply what is primary structure? It is the sequence of amino acids from N terminus to C terminus. That's it. It is called as primary structure. But this primary structure is not always present as a linear one. It is having different foldings and all. That's why it is able to form secondary, tertiary and quaternary structures like this. And if we are observing the secondary structure, different types of secondary structures we are observing in different portions of a protein. Read the NCERT sentence very carefully. Many students, what they are thinking is, in one protein only one secondary structure is present. It is not like this. It is not like that. Please understand this. One protein, within a single protein, in different portions, different secondary structures can be present. Sometimes complete protein may have same secondary structure. But in some cases, in a single protein, in different portions, different secondary structures are possible. This is very important point. Many students are not focusing on this point, so I am repeating this. So in this way, different types of secondary structures we are observing in proteins. What are these? So some proteins, they are having some proteins in some portions, see here, in some portions they are having some helical structures. It is called as alpha helical structure. More details were not given in NCERT, that's why I am not going into further details. But see here, the same protein, continuation here. At some portions, it may be having some beta pleated sheets. And these beta pleated sheets, they may be present in parallel manner or they may be present in anti-parallel manner. So in this way, we are talking about different types of these particular secondary structures. But please remember, alpha helices always in the proteins we are observing only right-handed helices only. In the protein always we are observing right-handed coiling of this particular protein backbone we are talking. Clear? And next one regarding the beta sheets, if you are observing. In the some case of beta sheets, what happens is, in that particular portion, for all these portions, N terminus on one side, C terminus on other side. This type of beta pleated sheet is called as parallel beta sheet. But in some cases, what happens is, they are present in opposite directions. And if this particular stretch is present, this particular portion is called as anti-parallel beta sheet. So in this way, different types of secondary structures are possible. Clear? And now we are going with a unit introduction. Just in the next page of that unit introduction, they have given about the G.N. Ramachandran. Yes or no? Scientist name. Now we are talking about that particular scientist only. Now here, that scientist, here they have given, his discovery, his means whose? It is the discovery of this, discovery by this G.N. Ramachandran. So G.N. Ramachandran discovered what? Triple helical structure of collagen. This sentence was given in the NCERT, and this is the triple helical structure of collagen. Triple helical structure of collagen is also an example for secondary structure of protein. Please remember, for secondary structure of protein, what are examples we are studying? Alpha helix and beta sheets you are studying. And triple helical structure of collagen is also an example for secondary structure. Please observe this very carefully. Clear? Next one, this is one important point. And next one, this scientist has given a plot, and that plot is called as a Ramachandran plot based on his name only. His name is Ramachandran, so he gave a plot. That plot is called as a Ramachandran plot. This is the Ramachandran plot. And you don't need to worry about this particular plot, but what is the significance of this particular plot? So using this plot, if you know simply the sequence of amino acids in a protein, once again I am repeating, if you know the sequence of amino acids in a protein, in which portion, what type of secondary structures are present, what type of secondary structures can be formed, you can easily predict with the help of this particular plot. Please understand this. Once again I am repeating, if you know the sequence of amino acids in a protein, what are the different types of secondary structures that are possible in that particular protein, that you can easily guess, based on this Ramachandran plot. That's why this plot is very, very important in structural biology. Those scientists who are doing, they are trying to study the structures of protein, definitely they will use this particular plot. It is that much important. Clear? And in this way, this is another important point that you need to understand. And finally, at the end of this paragraph, they have given another sentence. What they are saying is, Ramachandran met Linus Pauling. Already in the chemistry you studied about Linus Pauling, so please remember this. Linus Pauling, and was deeply influenced by his publications on models of the alpha helix and beta sheet structures that directed his attention to solve this structure of collagen. So now what is the conclusion here? Linus Pauling already did some research on alpha helix and beta sheets. This is another important point. Alpha helix and beta sheet structures. So who is the scientist related to this? It is the Linus Pauling. So he met him and he was influenced by this, and ultimately that particular information was useful in solving the structure of collagen, and based on this he proposed triple helical structure of collagen. So please understand this. So that's why Linus Pauling was actually working with alpha and beta sheets of this particular protein structure, and Ramachandran was related with triple helical structure of the collagen. And along with this, Ramachandran plot is very important to know possible secondary structures in a protein. This is what the important information related to this information from this particular scientist page. Remaining information is not needed. Simply these are the important points. Observe them. With their secondary structure of protein is completed. Now we are going with the tertiary structure of protein. And now what happens? Primary structure will fold and form secondary structure, and it will undergo further folding and form secondary tertiary structure. And now it is generally making some random coiling like this. It is coiled like this, so that it is making like some woolen ball and all. So in this way what happens, the amino acids presented at different distant locations also can be coming very closely, and in this case we are observing some gaps, and generally they are called as crevices or pockets, and they may be involved with some various functions. In this way, proteins are having this tertiary structure, and this tertiary structure you can consider as a three-dimensional view, and majority of the proteins, for their normal biological functions, they should compulsorily have this tertiary structure. This is what they are saying here. Next one. So this is what about the tertiary structure of a myoglobin. One random example I am giving you, that's it. Next one is regarding quaternary structure, and please observe the concept between tertiary and quaternary structure. Many students are not getting clear difference here. Please remember here, what is the difference between here tertiary and quaternary structures? Observe the concept here. Let us take one protein. And suppose this protein is folded like this. So for this single protein, the highest level of conformation is tertiary. That's it. Can single protein show tertiary quaternary? No, it cannot show. Then when you will see the quaternary, when two or more are interacted. Observe carefully. Suppose this is one protein. For this single protein, what is the highest level? Tertiary. For this single, it is tertiary is the highest. Now let us see another one. Suppose this is the another one. For this one, what is the highest level? Tertiary. Observe carefully. For this, it is tertiary. For this also, it is tertiary. Individually, single proteins will have tertiary as the highest. That's it. They cannot have quaternary. Then when you will see the quaternary, if two proteins are interacted, maybe what type of interaction? They may be through some metal ion or something. If two different proteins are interacted, now this overall structure is called as quaternary. Observe carefully. Understood or not? Individual protein will have tertiary as the highest level of conformation. When two or more means what? 3, 4, 5, 6. Likewise, if many are interacted, now this overall will be called as quaternary. Understood or not? This is the important point that you need to notice. So in this way, within the quaternary structure also, individually each protein is having tertiary as the highest. Suppose you are talking about hemoglobin as example. In the hemoglobin, why you are taking hemoglobin example here? 1, 2, 3, 4. 4 proteins are interacted. Out of these 4, for this one, what is the highest? Tertiary. For this, what is highest? Tertiary. For this, what is highest? Tertiary. For this, what is highest? Tertiary. So for individual protein, if you ask, tertiary. But all these are interlinked with each other like this. All these are interlinked. Once all these are interlinked, all this combinedly it is quaternary structure, because two or more are present. Yes or no? In this case, what? More than two are there. So that's why it is quaternary structure. So in this way, if many are interacting with each other, then only you will see quaternary structure. If only one protein is there, highest will be tertiary only. Don't make mistake. Many students are not understanding this logic. So in the case of human hemoglobin, what are there? So two alpha chains are there, two beta chains are there. Total how many are there? Four are there. All these are interacted. That's why you are observing quaternary. Suppose you are talking for myoglobin, single protein. Myoglobin is having single protein, so it will have highest as tertiary only. Myoglobin cannot have quaternary, but hemoglobin is having quaternary. Understood or not? So in this way also questions can be asked. Observe carefully. Anyhow, this is not related to this chapter. Observe carefully. So hemoglobin adult type, hemoglobin fetal type. This information is important. So we will discuss in different other chapters. Fetal hemoglobin is having two alpha chains and two gamma chains. Whereas adult hemoglobin is again two types, adult 1 and adult 2 type. So generally what we are studying is adult 1 type. Hemoglobin adult 1 type, it is having two alpha chains, two beta chains. Hemoglobin adult 2 type is having two alpha chains, two delta chains. Okay, so in this way, but majorly we are studying about adult 1 type, it is alpha to beta 2, two alpha chains and two beta chains. This is what generally we are studying. Clear? With this, amino acids and proteins information is completed. Now we are going with the lipids. Now let us quickly go through this. Generally these are water insoluble. They are generally not soluble in water, and that's why they are hydrophobic. So they are not soluble, that's why they are hydrophobic. Next one, they could be like some simple fatty acids. So under the lipids, simple fatty acids can be considered as lipids, and even some glycerol is also considered as lipid. These are some simple molecules. Not only this, many complicated molecules are also present in this. First we will talk about the fatty acid. So what are these fatty acids? Fatty acids, they are having one COOH, carboxylic acid group, and then there is one hydrocarbon chain is present, and this may be saturated, and in some cases this may be unsaturated also. It may be anything. So in this way, it is the carboxylic acid group that is attached to an R group. That R group can be anything. This R group can be a simple methyl group, ethyl group, or even higher number. So in the range of 1 to 19 carbons. Only for the R group, if you see 1 to 19, not only 19, it can be more also, but at your level you are focusing up to this one only. And now they have given an example called palmitic acid. This is having 16 carbon. Please remember 16 carbons including COOH group. If you are removing COOH, then how many will be there? 15 carbons are present. Observe the content of NCRT clearly. Whereas in the case of arachidonic acid, if you are observing, total how many are there? 20 carbons are there. Whereas R group is having 19 carbons. COOH is having 1 carbon, R group is having 19 carbons. But in palmitic acid, COOH is 1 carbon, R group is having 15 carbons. Observe the content carefully. So in this way, some examples they give. This is some list of some fatty acids, but you don't need this entire list. Observe this palmitic acid. It is having 16 carbons. 0 means what? It is not having any double bonds. No double bonds. 0 double bonds. That means it is saturated. That's why it is written as CH3, CH2 14 times, COOH. In NCRT also clearly they mentioned. Observe this carefully. If you are talking about arachidonic acid, it is having overall 20 carbons. Though it is having 20 carbons, now here it is having total 4 unsaturations. What are the positions of unsaturations? Double bonds. At 5th carbon, 8th carbon, 11th carbon and 14th carbon, you are finding double bonds. Understood or not? These are the positions of double bonds. So total 4 double bonds are present. That means, as per NCRT, if you observe, palmitic acid they have mentioned it is saturated fatty acid because there are no double bonds, triple bonds. But arachidonic acid is having double bonds. So in the double bonds also, in the structure, if only one double bond is there, that is called as a mono unsaturated fatty acid. Mono unsaturated fatty acid, it is called as mono unsaturated fatty acid. If more than one double bonds are there, it is called polyunsaturated fatty acid. So if you are observing this arachidonic acid, more than one double bonds are there. Total how many? 4 are there. That means more than one are there. So more than one are present means arachidonic acid. Arachidonic acid is example for polyunsaturated fatty acid. Now here saturated fatty acid. For this, palmitic acid is example. These two are NCRT names. You should compulsorily know the information for at least NCRT names. Okay. This is what the information. Remaining names are optional, so leave it. Next one, another simple lipid, that is about the fatty acid. Another simple lipid is the glycerol. Its structure is CH2OH, CHOH and CH2OH. So 3 carbon containing. So hydroxy, hydroxy, hydroxy. 1, 2, 3 trihydroxy propane is nothing but glycerol. Like this also you can write 1, 2, 3, 1 comma 2 comma 3 trihydroxy propane is the glycerol. Understood or not? This also you can consider as a lipid. But generally what we are observing is fatty acid and glycerol are interlinked with each other. So this is the glycerol. This is the fatty acid. Any fatty acid, any random example you can take. But here also 3 carbons are there. If one fatty acid is only attached, it is called as monoglyceride. If 2 fatty acids are attached to the glycerol, then it is called diglyceride. If to all the 3 carbons, 3 fatty acids are attached, then it is called as a triglyceride. So these are also considered as lipids. But here what is the bond between fatty acid and glycerol? Fatty acid COOH group, glycerol OH group. Between COOH acid and OH alcohol, between acid and alcohol, what is the group? What is the bond that is formed? It is the ester bond. It is the ester bond. That's why they use the sentence called as fatty acid is esterified to glycerol. This is how they will say. Found esterified to with glycerol, right? So what is that esterified? Ester bond is formed between fatty acid and glycerol. Fatty acid and glycerol. This is what the point is. During this process, anyhow, water molecule will be lost. So this is one another important information that you need to understand. Then they are talking about fats and oils. So what is the anyhow they are lipids only. But what is the difference between fats and oils? Now this is very important. Oils will have lower melting point, whereas fats will have high melting point in the normal condition. So please remember, generally at room temperature. So in the scientific names, in the scientific terminology, if you are observing, what is room temperature means? So suppose you are normally observing a temperature in the room. If you are giving definition like this, in different conditions like in winter and monsoon and in the summer, room temperature will change. But scientifically, if they are saying at room temperature, the meaning is 25 degrees centigrade. At room temperature means 25 degrees centigrade. So at the room temperature, if it is present in the liquid form, it is called as oil. With respect to the lipids we are talking, the lipid that is present in the liquid form at room temperature is called as oil. If the lipid is present in the solid form at room temperature, it is called as a fat. So in this way, based on this, what you can say, oils will have lower melting point and fats will have higher melting point. This is what the conclusion that they are giving like this. Understood or not? So in this way, the gingerly oil, even in the winter also, it is like oil, it is in the liquid form only. So that's why you can consider this as oil. Suppose you are talking about the ghee or vanaspati, they are present as solid and you can consider them as the fats. So these are some standard examples that you can understand. Next one is regarding the phospholipids. So till now we studied about monoglyceride, diglyceride, triglyceride, right? In the diglyceride, what happens? Two fatty acids are attached to, in the diglyceride what happens? Two fatty acids are attached to the glycerol. Now here, two fatty acids are attached to the glycerol. If the third one is attached to phosphate group, then it is called as a phospholipid. So in the phospholipid, phosphate group is attached, and generally these are the diglycerides having the phosphate group. And even this phosphate group is also further substituted with some other molecule. And here if you are observing in NCRT, they have given the example of the lecithin. So in the lecithin, what it is called as? Phosphatidyl choline. So this is the choline molecule that is attached to this. So that's why it is called a phosphatidyl choline. So phosphatidyl choline is also called as lecithin. In the same way, anyhow, these names are not there. Different other molecules called phosphatidyl ethanolamine, also called as cephalin. You can find it, and phosphatidyl serine is there. So in this way, different types of phospholipids are present, and generally they are found in cell membranes, plasma membranes. No, plasma membrane, not only plasma membrane, even the membranes of organelles also, they are having this type of phospholipids. Understood or not? This is what they are talking here. And not only this, in the case of neural tissues, especially if you are observing, some more complex type of lipids are observed. And there we are generally observing sphingolipids, cerebrocytes, gangliosides. So these are different types of some more complex lipids. But this information was not given in NCRT, so I am not going into those details. Clear? And next, another interlinked point is, so in the case of neural system, we studied about some white matter and all. So white matter is mainly having what? So myelinated neurons are present. What in this myelin? So myelin sheath in the central nervous system is produced by oligodendrocytes. Myelin sheath in peripheral nervous system is produced by Schwann cells. Already we studied about that information. And that myelin sheath, generally it is having what? 80% lipids and 20% proteins. And in that 80% protein lipids also, so now here the major type of lipid is galactocerebroside. So that's why generally we say that the myelin sheath is also a type of lipid molecule. Mainly it is a lipid molecule, yes or no? So this is what some interlinked information. If possible, try to remember this information also. Clear? So this is what the NCERT example that they have given. So this is the fatty acid example. Palmitic acid, it is having total 16 carbons and it is saturated fatty acid. Next one, glycerol. It is 1, 2, 3 trihydroxypropane. You can consider this as some simple form of lipid. And here some type of lipids. One fatty acid may be attached, 2 may be attached, 3 may be attached. Here it is the 3 attached. That's why it is triglyceride example. Clear? Next one, phospholipid. Less than example they have given here. And here even cholesterol, sterols also. Sterols are also coming under a type of lipids. So here it is the cholesterol. So already regarding the cholesterol and its role in the case of animal cell membranes, we studied in the cell chapter also. Clear? So with that, that lipid spot is also completed. But please remember, lipids are not polymers. So they are not true polymers. They are non-polar in nature. They are hydrophobic in nature. They are acid insoluble. That's why they are grouped together and coming under retentate or acid insoluble fraction. Clear? With that it is over. Now we are going with the nucleotides and nucleic acids. So nucleic acids are also macromolecules. They are true macromolecules. They are polymers of nucleotides. That's why they are called as polynucleotides. And now here their building blocks. Each unit is called as nucleotide. And each nucleotide is having 3 components. What are the 3 components of nucleotide? Nitrogenous base and a sugar. What is that sugar? Either ribose. Ribose in the case of RNA, 2-deoxyribose in the case of DNA. Yes or no? And nitrogenous bases are heterocyclic compounds. They are of different types, purines, pyrimidines, right? Slowly we will study those. And along with nitrogenous base and sugar, there is another one that is called as phosphate group, also called as phosphoric acid, and more clearly it is called as orthophosphoric acid. It is called as orthophosphoric acid. And here first we will see about the nitrogenous bases under the nucleotides. Nitrogenous bases, again based on the heterocyclic rings that are present, they are of 2 types. If the basic purine ring is present, they are called as purines. If the basic pyrimidine ring is present, they are called as pyrimidines. And here under the purines we are studying adenine and guanine. Adenine and guanine are the substituted purines. Adenine is 6-amino purine, guanine is 2-amino 6-oxypurine. Whereas cytosine, uracil and thymine are pyrimidines. They are substituted pyrimidines. Cytosine is 4-amino 2-oxypyrimidine and uracil is 2,4-dioxypyrimidine and thymine is 5-methyl 2,4-dioxypyrimidine, also called as 5-methyl uracil. All these names are not needed, but at least thymine is called 5-methyl uracil mentioned in NCERT, so please try to remember this. Clear? Next one. In the case of RNA, if you are talking, now what we are observing, in the case of RNA, nucleotides are present. Now all the 3 are combined. Now what happens? Nitrogenous base, if it is combined with sugar, nitrogenous base plus sugar is called as nucleoside. And to this nucleoside, if a phosphate group is attached, then it is called as nucleotide. Please remember these names. What is nucleoside? Nitrogenous base plus sugar. But what is nucleotide? Nitrogenous base plus sugar plus phosphate group is called as nucleotide. Now again here, in the case of RNA and DNA, what are the various names that we are using? These names were clearly mentioned in NCERT, even in class 11th and class 12th. So you should compulsorily remember these names. Adenine, if combined with ribose, it is called adenosine. Guanine combined with ribose is called as guanosine. Cytosine plus ribose is called as cytidine. And uracil plus ribose is called as uridine. So please remember these names here. For the nucleosides, with respect to purines, osine is coming at the end. For the nucleosides, with respect to pyrimidines, idine is coming at the end. So please remember this. So this is what the names we are writing. And then to this, if phosphate group is attached, phosphate means orthophosphoric acid, ic acid. So now how they are changed? Now they will become corresponding ileic acids. Corresponding ileic acids will come. Adenosine plus phosphoric acid, it is adenylic acid. Guanosine plus phosphoric acid, guanylic acid. Cytidine plus phosphoric acid, cytidylic acid. Uridine plus phosphoric acid, uridylic acid. So in this way, if you are observing the RNA, what type of nucleotides are present? These are the nucleotides that are present. But please remember, in the case of RNA, thymine is not present, and the spelling of thymine is very important. Some students are not noticing the difference also. Here there are two names. One is called thymine, 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 and other one is called THIAMINE, thiamine. THIAMINE, thiamine. This is thymine. And this THYMINE, thymine is a nitrogenous base, pyrimidine. But THIAMINE is vitamin B1, vitamin B1. So spellings are very important for these two things. So don't make any mistake. Clear? Next one. Next one. With respect to DNA, if you are talking, in the DNA, adenine, guanine, cytosine are also present, but uracil is present in RNA, and in the case of DNA, thymine is present. Understood or not? This is what one important point that you need to notice. Next one, here, in the case of DNA, it is not the normal ribose sugar, it is the 2-deoxyribose sugar that is present. And now once you are adding this, now what happens? Observe carefully. Adenine plus 2-deoxyribose is 2'-deoxyadenosine. Please remember, when you are writing individual sugar, then it is 2-deoxy. But once adenine and that means once nitrogenous base and 2-deoxyribose are attached, now what you need to do? You need to give the primes for the carbons that are present within the sugar. That's why now it will become 2'-deoxy, and here also it will become 2'-deoxy. And this explanation I gave very clearly in the regular lectures. So those who don't know why we are keeping primes here, so you please go through that regular lecture. Clear? Now let us continue further. Adenine plus 2-deoxyribose is 2'-deoxyadenosine. Guanine plus 2-deoxyribose is 2'-deoxyguanosine. Cytosine plus 2-deoxyribose is 2'-deoxycytidine. Thymine plus 2-deoxyribose is 2'-deoxythymidine. Clear? And now you need to add the phosphoric acid. So now what will be formed? 2'-deoxy adenylic acid, 2'-deoxy guanylic acid, 2'-deoxy cytidylic acid, 2'-deoxy thymidyllic acid. That's it. So in this way, these are the nucleotides that are present within the DNA. So in this way, nucleic acids are polymers of nucleotides. So in this way, we studied individual components. Next one, nucleic acids like DNA and RNA consists of nucleotides only. They are made up of only nucleotides. Next one, DNA and RNA functions as genetic material. This sentence is very important. DNA is genetic material in all 5 kingdoms plus some viruses, whereas RNA is genetic material in only few viruses. That's it. Clear? This is what another important point we need to understand. This is what the NCERT diagram is, and here they have given the structure of adenine as an example for purine. This is the structure of uracil as an example of pyrimidine. Now here what they did, adenine plus this is the ribose sugar, adenine plus ribose, that's why it is adenosine. This is uracil plus ribose, that's why it is uridine. So it is these two are example for nitrogenous bases, these two are examples for nucleosides. And here what they did, nitrogenous base adenine plus ribose sugar plus and phosphate group means it is nucleotide. This is adenylic acid. So in this way they have given few examples, but you need to understand all these things with structures, names, everything. In the regular lecture we studied with very clear details, and this information is important. Biomolecules of class 11th and also molecular basis of inheritance of class 12th. So in both the chapters it is very important. So you should compulsorily understand all these things with structures, and at least names are important. Nitrogenous bases names are important, nucleosides names are important, nucleotides names are important with respect to RNA and with respect to DNA both. Clear? And with this part 1 of biomolecules part is completed. And before concluding this, once again I am telling you that these revision video lectures are available on our Live Prep app and also on Live Prep India YouTube channel, but the test series is available only on the app. Clear? So with this part is completed. So in the next lecture we will meet with remaining part of the biomolecules. Thank you.